Welcome to For Science Salah Lotfy channel, where science meets curiosity. في هذه القناة لا نكتفي بعرض الأبحاث العلمية، بل نترجمها إلى المعرفة التي تصل إليك أينما كنت. Our mission is to make global scientific research understandable, accessible, and multilingual. جميع الفيديوهات العلمية هنا مترجمة إلى اللغة العربية، بالإضافة إلى لغات عالمية أخرى. French, Spanish, German, Urdu, and more. Because science speaks every language. نعم، العلم يتحدث بكل اللغات، ونحن نحرص أن يصلك بلغتك. If you're passionate about science, you're in the right place. اشترك الآن وابدأ رحلتك مع العلم خطوة بخطوة. For Science Salah Lotfy channel, العلم للجميع.
Welcome to For Science Salah Lotfy channel, where we explore cutting-edge research shaping the future of science and technology. Today, we highlight a powerful scientific contribution in the fight against a hidden but pervasive threat: environmental pollution caused by residual antibiotics in our wastewater. These life-saving drugs enter our water systems daily, not just from pharmaceutical manufacturing, but primarily through human and agricultural waste. The problem is, conventional wastewater treatment plants are often ill-equipped to filter out these complex and resilient molecules. This persistence in the environment represents a serious challenge with a dual impact. It threatens aquatic life by disrupting delicate ecosystems. More alarmingly, it creates the perfect breeding ground for one of the greatest public health crises of our time: the rise of antibiotic-resistant bacteria, or superbugs. Finding an effective solution is therefore a global health imperative. In a groundbreaking study published in ChemistrySelect, researchers present an innovative and eco-friendly solution. Their approach centers on creating advanced nanocomposites from readily available natural materials: sodium alginate, a polysaccharide from brown seaweed, and nano-bovine serum albumin, a protein formed into incredibly small particles. The key to their method is gamma irradiation, a clean and highly effective technique. This process forges strong, stable bonds between the components without needing chemical additives, which can introduce secondary contaminants. This synthesis method is not only efficient but also aligns with green chemistry principles. The gamma radiation triggers a process called cross-linking, creating a robust three-dimensional hydrogel network. This structure gives the material its smart properties. When placed in water, these hydrogels demonstrate an extraordinary capacity for swelling, expanding up to an astonishing 4,400% of their original size. This massive expansion is crucial. It dramatically increases the surface area and exposes more active sites within the nanocomposite, making it a highly effective sponge for pollutants. To validate their creation, the team tested the material against a common and persistent pollutant, the antibiotic ceftriaxone sodium. Under optimized laboratory conditions, the results were outstanding. The material successfully removed an impressive 90% of the antibiotic from the water sample, confirming its powerful adsorption capabilities and proving it can effectively extract these harmful pharmaceutical residues. Our detailed analysis reveals a fascinating story. Kinetic studies confirm the adsorption process follows a pseudo-first-order model, meaning the initial uptake of pollutants is incredibly fast, driven by the vast number of available active sites on the nanocomposite. Furthermore, the isotherm analysis perfectly aligned with the Freundlich model. This is significant because it indicates a heterogeneous surface and supports multi-layer adsorption. Essentially, pollutant molecules don't just form a single layer, they stack up, dramatically increasing the material's purification capacity. These findings represent a major leap forward. We have demonstrated a low-cost, scalable, and environmentally friendly method to remove hazardous pharmaceutical residues. This technology holds immense potential for safeguarding our water supplies, preventing the spread of antibiotic resistance, and protecting fragile ecosystems. Future research will focus on scaling up production and conducting pilot tests in real-world wastewater streams, paving the way for a cleaner, healthier future. Stay tuned for more scientific breakthroughs that matter. Welcome to For Science Salah Lotfy channel. In today's episode, we spotlight a remarkable study that addresses one of the urgent challenges facing our environment: the removal of antibiotic pollutants from wastewater. The research, titled Adsorptive Removal of Residual Antibiotics Pollutants from Waste Solutions Using Gamma-Induced Preparation of Nanocomposites, was recently published in ChemistrySelect 2025. This work was conducted by an outstanding team from the Egyptian Atomic Energy Authority, National Center for Radiation Research and Technology, Polymer Chemistry Department, including Associate Professor Shaimaa Mounier Nasef and Dr. Mai Mostafa Mohamed, Professor Dr. Ghada Adel Mahmoud, and Professor Dr. Tarek Mansour Mohamed, who served as the corresponding author. Their study focuses on preparing innovative nanocomposites by combining sodium alginate with nano-bovine serum albumin using gamma irradiation. These nanocomposites act as powerful adsorbents for antibiotics in wastewater. The approach offers a sustainable, low-cost, and environmentally friendly solution to eliminate pharmaceutical residues that pose significant risks to human health and ecosystems. This important research opens new pathways for wastewater treatment technologies, where advanced radiation chemistry and nanoscience come together. Stay with us on For Science Salah Lotfy channel, as we continue to present pioneering scientific studies that shape a cleaner, safer future. Welcome to For Science Salah Lotfy channel. In our modern world, we are surrounded by invisible threats but also protected by unseen shields. We trust our infrastructure, our medicine, and the systems designed to keep us safe. But what happens when one of our greatest medical triumphs begins to fuel a silent, creeping crisis hidden in the very water we depend on? Today, we dive deep into one of the most critical environmental challenges of the 21st century and explore a pioneering scientific solution that offers a glimmer of hope. Our focus is on a specific, insidious form of pollution: the contamination of our wastewater with antibiotics. These miracle drugs, designed to save lives, are inadvertently creating a new danger. We'll investigate how this happens and reveal an innovative nanocomposite, a material engineered at the atomic scale, developed to solve this growing problem before it spirals out of control. The pollution of our global water systems with residual antibiotics is not a future threat. It is a present-day reality, and one of the most pressing environmental issues of our time. These compounds are classified as contaminants of emerging concern, meaning their presence is widespread, but the full extent of their ecological and health impacts is only now being understood. And what we are learning is deeply alarming. Conventional wastewater treatment facilities, marvels of engineering though they are, were never designed to filter out these complex and persistent pharmaceutical molecules. To grasp the scale of the issue, we must first look at the source. Antibiotics are cornerstones of modern medicine and agriculture, widely used to treat bacterial infections in both humans and animals. Global consumption is immense, measured in hundreds of thousands of tons annually. But here's the fundamental problem. These powerful drugs are designed to be stable. When we ingest them, they are not fully metabolized or broken down by our bodies. A significant fraction, sometimes up to 90%, is excreted through urine and stool, still chemically active. From our homes, hospitals, and farms, this waste flows into municipal sewer systems. Once inside the treatment plants, the antibiotics create a low concentration but constant chemical pressure on the vast populations of bacteria present in the sludge. This environment becomes a perfect, high-speed training ground for antibiotic-resistant bacteria, often called superbugs. The non-resistant bacteria are killed off, while the few that naturally have or develop resistance survive, thrive, and share their resistance genes with other bacteria. The treated water, now a cocktail of antibiotic residues and newly empowered superbugs, is then discharged back into our rivers, lakes, and oceans, contaminating the very sources of our drinking water and entering the food chain. This is no longer just pollution, it's the active cultivation of a global health emergency. The constant exposure to antibiotics in wastewater creates a high-pressure evolutionary crucible for bacteria. Here, they don't just multiply, they actively share survival tools. Through a process called horizontal gene transfer, bacteria pass resistance genes to one another using tiny packages of DNA called plasmids. This allows resistance to jump between different bacterial species, accelerating the rise of multi-drug-resistant organisms, or superbugs, like MRSA and CRE. These pathogens render our most critical medicines useless, turning common infections into life-threatening crises. The threat isn't confined to hospitals. When these resistant bacteria spill into our ecosystems, they contaminate soil and water, disrupting delicate microbial balances. Wastewater treatment plants designed to protect public health ironically become hotspots for this dangerous genetic exchange, breeding new forms of resistance that threaten the entire planet. The spread of antibiotic resistance is a silent, pervasive cycle. It begins when treated or untreated wastewater, laden with resistant bacteria and residual antibiotics, is discharged into rivers and lakes. This contaminated water is then often used for agricultural irrigation, seeping into our soils and watering the crops we eat. Livestock may drink directly from these water sources, ingesting both the resistant bacteria and the antibiotics. The bacteria can then colonize the animals, and the antibiotics can accumulate in their tissues. From there, the path back to humans is frighteningly direct, through the consumption of contaminated meat, produce, or even drinking water. This intricate web of connections is what scientists call a One Health problem, where the health of humans, animals, and the environment are inextricably linked. A failure in one domain inevitably triggers a crisis in the others, creating a global feedback loop that makes these superbugs more prevalent and far more dangerous. Faced with this escalating crisis, a forward-thinking group of researchers recognized that simply trying to kill the resistant bacteria wasn't enough. The root of the problem is the selective pressure exerted by the antibiotics themselves. As long as these compounds persist in the environment, they will continue to drive the evolution of resistant strains. The true challenge, they proposed, was to intercept and remove the antibiotics from wastewater they can cause harm. Their objective was ambitious, to synthesize a novel adsorbent material. But it couldn't be just any material. To be a viable global solution, it had to meet 3 critical criteria. First, it needed to be low cost, ensuring accessibility for communities worldwide. Second, it had to be highly efficient, capable of capturing a wide range of antibiotic molecules, even at trace concentrations. And finally, it had to be fundamentally eco-friendly. The solution to pollution could not become a pollutant itself. Their innovative solution was a sophisticated hybrid nanocomposite, engineered from the ground up. The team strategically selected 3 core components. The foundation is sodium alginate, a natural polysaccharide from brown algae. It's safe, biodegradable, and forms a porous, gel-like hydrogel, the perfect scaffold. To amplify its capabilities, they incorporated acrylic acid. This monomer was grafted onto the alginate backbone, vastly increasing the number of carboxyl groups, which act like molecular-scale magnets for antibiotic molecules, dramatically boosting the material's adsorption capacity. The final clever component is nano-sized bovine serum albumin, or BSA. BSA is a common protein with a complex, folded structure containing hundreds of amino acid residues. This structure gives it an incredible, almost tailor-made ability to bind with drug molecules. By reducing it to the nanoscale, the researchers maximized its surface area, making it exponentially more effective. To fuse these components into a stable structure, they employed gamma radiation. This clean energy technique initiates polymerization and cross-linking, creating the robust hydrogel network without toxic chemical initiators. The result is a synergistic masterpiece, a low-cost, biodegradable sponge where the alginate provides the frame, the acrylic acid adds countless hooks, and the nano-BSA acts as a high-affinity trap, working in concert to pull antibiotic contaminants out of water. The creation of these advanced nanocomposite hydrogels begins with a sophisticated, clean manufacturing process. Instead of using chemical initiators that can leave harmful residues, researchers employed gamma radiation. This single-step method initiates 2 crucial reactions: polymerization and cross-linking. When the aqueous solution of polymer building blocks and nanoparticles is exposed to gamma rays, the energy splits water molecules into highly reactive free radicals. These radicals act as catalysts, triggering the building blocks to link into long polymer chains. Simultaneously, they induce cross-linking, stitching the chains into a robust 3-dimensional network. This intricate structure is the essence of a hydrogel, ensuring the nanoparticles are uniformly distributed and securely entrapped. Once synthesized, the hydrogel's effectiveness hinges on its precise internal architecture. To verify this, researchers used powerful analytical techniques. Fourier transform infrared spectroscopy, or FTIR, analyzed the chemical bonds, confirming successful polymerization and the chemical integration of the nanoparticles. Next, scanning electron microscopy, or SEM, provided high magnification images of the physical structure. These revealed a highly porous, interconnected network, a microscopic sponge vital for creating a massive surface area. SEM also confirmed the even dispersal of nanoparticles. Finally, X-ray diffraction, or XRD, probed the material's crystalline structure, confirming the nanoparticles were present in their desired form. Together, these analyses painted a complete picture of a hydrogel engineered for its task. A remarkable property of these hydrogels is their incredible capacity for swelling. Researchers observed a swelling ratio of 4,400% in just 6 hours. meaning the hydrogel can absorb 44 times its own dry weight in water. A small dry speck transforms into a large swollen gel. This phenomenal swelling is driven by water-loving hydrophilic groups on the polymer chains. These groups attract water molecules into the hydrogel's interior, while cross-links prevent the structure from dissolving. This process is fundamental to the hydrogel's function. As the network expands, it opens up, making the embedded nanoparticles more accessible to contaminants, effectively unfurling the molecular net for capture. With its structure confirmed, the hydrogel was tested against a real-world pollutant, ceftriaxone sodium, a powerful antibiotic whose persistence in wastewater drives antibiotic resistance. The results were exceptional. The nanocomposite hydrogel achieved an adsorption efficiency of 90.2%, removing the vast majority of the dissolved antibiotic. This process, known as adsorption, involves the antibiotic molecules adhering to the vast internal surface of the hydrogel, binding to active sites on the embedded nanoparticles. This high efficiency is a significant breakthrough, showcasing performance that can surpass many traditional water treatment methods. It confirms the hydrogel is an effective and selective trap for hazardous pharmaceutical waste. To optimize this technology, it's crucial to understand, and it works. Kinetic studies revealed the adsorption process follows a pseudo-first-order model, indicating a very rapid initial capture of antibiotics, limited mainly by available binding sites. Thermodynamic analysis provided deeper insights. The process is spontaneous, meaning it occurs naturally without needing external energy, a critical feature for passive water treatment. Furthermore, the process was found to be endothermic. This fascinating discovery means the reaction absorbs heat, so the hydrogel's efficiency actually increases at slightly higher temperatures. This suggests a complex binding mechanism and opens possibilities for optimizing performance in industrial settings where wastewater might be warmer. The significance of this research lies in its combination of scientific novelty, environmental safety, and practicality. The nanocomposites are low-cost, biocompatible, and highly effective. Imagine a future where wastewater treatment plants worldwide integrate such nanocomposite materials. The result would be safer water, healthier ecosystems, and reduced antibiotic resistance. This study highlights the power of science to tackle urgent global challenges. From the lab bench to environmental protection, innovations like these remind us that solutions are within reach. Stay with us on For Science Salah Lotfy channel as we continue exploring groundbreaking scientific research that shapes a cleaner and healthier future for all.
Welcome back to For Science with Salah Lotfy. Our modern world depends on chemicals, but what happens after we use them? Many, including powerful antibiotics, end up in our wastewater. Conventional treatment plants can't always remove these microscopic contaminants, fueling a silent crisis, the rise of antibiotic-resistant bacteria in our environment. This poses a major threat to global health. But what if we could design a smart sponge to specifically soak up these dangerous residues? Today, we're exploring groundbreaking research that offers a potential revolution in how we clean our water. The solution lies in a remarkable material, a nanocomposite hydrogel. You might know hydrogels from things like contact lenses. They're polymer networks that are fantastic at absorbing water. But these scientists took it a step further. By embedding functional materials at the nanoscale, they created a nanocomposite hydrogel, a super absorbent specialized tool designed to trap and remove specific antibiotic residues from water. So what are the ingredients for this molecular sponge? The researchers started with 4 key components. First, sodium alginate, a natural polymer from brown seaweed, which forms the main scaffolding. Next, acrylic acid, a synthetic compound that helps build a strong, cross-linked network. For the nano part, they incorporated bovine serum albumin, or BSA, a common protein from cow blood. And to test their creation, they used a target pollutant, the widely used antibiotic, ceftriaxone. Each material plays a crucial role. The alginate chains form the hydrogel's backbone. The acrylic acid acts like a chemical glue, linking these chains to create a robust, water-loving gel. The real magic, however, comes from the BSA protein. These protein molecules are dispersed throughout the gel, creating countless tiny specialized pockets. These pockets have the perfect shape and chemical affinity to recognize and latch onto the antibiotic molecules, effectively trapping them like a key in a lock. The first step in building this advanced material was to create the nano component. For this, the team turned to a common, readily available protein called bovine serum albumin, or BSA. They employed a clever technique known as the desolvation method. This begins by dissolving the BSA protein in a salt solution. Then, with precision and care, ethanol is slowly introduced into the mixture. As the ethanol concentration gradually increases, the protein molecules are forced to huddle together, self-assembling into incredibly small, stable spheres. The result? Perfectly formed nanoparticles, each measuring a mere 7 to 8 nanometers in diameter. To put that in perspective, you could fit over 10,000 of them side by side across the width of a single human hair. The team confirmed this minuscule size using powerful transmission electron microscopy, which allows us to visualize a world far beyond what light microscopes can see. With the nanoparticles ready, it was time to build their home, the hydrogel matrix. The researchers created a precursor solution by blending two key ingredients: alginate, a natural polymer extracted from seaweed, and acrylic acid, a monomer famous for its ability to form superabsorbent materials. Once thoroughly mixed, the freshly synthesized BSA nanoparticles were dispersed evenly throughout this viscous liquid. The final, crucial step was to solidify this mixture. Instead of using potentially toxic chemical crosslinkers, the team opted for a cleaner solution: gamma radiation. This high-energy source initiated a chain reaction, causing the polymer chains to link together, or cross-link. This process forged a robust interconnected network, effectively trapping the BSA nanoparticles within its pores. The final product is a true nanocomposite hydrogel, a highly porous, sponge-like material engineered to absorb enormous quantities of water, and with it, the pollutants we want to remove. With our hydrogels synthesized, it was time for testing. First up, swelling capacity. This is crucial because a hydrogel's ability to absorb water is directly related to its potential for capturing contaminants. More swelling means more surface area and more active sites. We placed a small, dry disc of our nanocomposite hydrogel into water, and the results were staggering. In just 6 hours, it swelled to over 4,400% of its original size, proving the nanoparticles were already enhancing its fundamental properties. But had we truly created a stable nanocomposite? We turned to a suite of powerful analytical tools. Techniques like Fourier transform infrared spectroscopy confirmed our chemical bonds, while scanning electron microscopy gave us an up-close look at the internal structure. The images revealed the nanoparticles were perfectly distributed and locked within the porous matrix. This is critical. We need the nanoparticles to stay put and not leach into the very water we're trying to clean. Now for the main event. We chose a stubborn pollutant, the antibiotic ceftriaxone. We prepared a solution mimicking contaminated wastewater, dropped in our hydrogel, and waited. To measure the outcome, we used a UV-vis spectrophotometer. It analyzes how much antibiotic remains by measuring how much light the water absorbs. The results were outstanding. Our nanocomposite hydrogel removed up to 90% of the antibiotic contamination. This is a huge leap forward, demonstrating a powerful and efficient new tool in our fight for cleaner water. This breakthrough represents a low-cost, eco-friendly solution that could be game-changing in fighting antibiotic pollution and resistant bacteria. That's today's scientific breakthrough. Don't forget to like, share, and subscribe for more cutting-edge research. Welcome to For Science Salah Lotfy channel. In a world brimming with technological marvels and medical breakthroughs, we often overlook the silent, invisible challenges that emerge in their wake. Today we delve into one such challenge, a consequence of our success in fighting disease, that now poses a profound threat to our environment and our future health. We are highlighting an innovative study that tackles one of the most pressing environmental and health challenges of our time, the pervasive contamination of our water systems with pharmaceutical pollutants. Specifically, we're talking about a problem that begins in our medicine cabinets and ends in our rivers and oceans: the removal of antibiotic pollutants from water. These miracle drugs, designed to save lives, are creating a slow-motion crisis once they are flushed into our wastewater. The groundbreaking research we are exploring is titled Adsorptive Removal of Residual Antibiotics Pollutants from Waste Solutions, using gamma-induced preparation of nanocomposites based on sodium alginate and nano-bovine serum albumin. A pivotal contribution to the field, it was recently published in the prestigious journal ChemistrySelect in 2025 Volume 10, under the publication number E2024066060. This work is proudly presented by Chemistry Europe, European Chemical Societies Publishing, and the renowned 2025 Wiley VCH GmbH, a testament to its scientific rigor and importance. This landmark study was carried out by a distinguished team of scientists from the Polymer Chemistry Department, a hub of innovation and advanced materials science. Their work is housed within the National Center for Radiation Research and Technology, under the umbrella of the Egyptian Atomic Energy Authority in Cairo, Egypt. This institution is at the forefront of applying nuclear and radiation sciences for peaceful purposes, from energy and medicine to, as we see here, critical environmental remediation. The brilliant minds behind this research are Associate Professor Shaimaa Mounier Nasef, Dr. Mai Mostafa Mohamed, Professor Dr. Ghada Adel Mahmoud, and the corresponding author, Professor Dr. Tarek Mansour Mohamed. For those in the scientific community wishing to engage with this work further, Professor Dr. Tarek can be reached at tarekmum75@yahoo.com. Their collective expertise has forged a powerful new tool for environmental protection. To truly appreciate the novelty of their approach, we must first understand our long journey with water purification. The quest for clean water is as old as civilization itself. Ancient Egyptians, as far back as 1500 BC, documented methods of clarifying water using sand and gravel filtration. The Romans engineered magnificent aqueducts that not only transported water, but often incorporated settling basins and filtration systems. For centuries, these physical methods, removing visible dirt and sediment, were the pinnacle of technology. The 19th century brought a paradigm shift. In 1854, British physician John Snow traced a cholera outbreak in London to a single contaminated water pump, proving that deadly threats could be invisible. This discovery ushered in the age of disinfection. By the early 20th century, cities began adding chlorine to their water supplies, a revolutionary step that drastically reduced waterborne diseases. Filtration and disinfection became the one-two punch of modern water treatment. But the challenges of the 21st century are different. Our water is now threatened by complex chemical micropollutants like antibiotics, which can slip right through these conventional systems. This is where a new scientific frontier comes into play: nanotechnology. Nanotechnology is the science of manipulating matter on an atomic and molecular scale, the realm of the nanometer, which is 1/1,000,000th of a meter. To put that in perspective, a sheet of paper is about 100,000 nanometers thick. By working at this incredibly small scale, scientists can create materials with extraordinary properties. One of the key principles is the explosion in surface area. A small amount of material, when broken down into nanoparticles, has a vastly larger surface area, making it incredibly reactive and efficient for applications like catalysis and, crucially, adsorption. In environmental science, nanotechnology has opened up a toolbox of solutions. Researchers have developed nanomembranes with pores so small they can filter out viruses and salts, photocatalytic nanoparticles that use sunlight to degrade organic pollutants, and metallic nanoparticles that can neutralize heavy metals. This field represents a monumental leap forward from sand filters and chlorine, offering targeted solutions for the invisible chemical threats of our modern world. And it is precisely this advanced approach that the team in Cairo has harnessed. This research focuses on a novel nanocomposite material created through a clean and elegant process: gamma irradiation. Unlike conventional chemical methods that can leave behind toxic residues, gamma irradiation is a form of pure energy. Think of it as a highly precise and powerful beam of light that initiates chemical reactions. In this case, it's used to stitch molecules together, creating a stable, three-dimensional structure without adding any new chemicals to the mix. It's a green chemistry approach to building advanced materials. The building blocks of this nanocomposite are brilliantly chosen for their properties and sustainability. The first is sodium alginate, a natural polymer extracted from the cell walls of brown seaweed. It's abundant, renewable, biodegradable, and completely non-toxic. In this composite, it forms the backbone, a sort of microscopic porous scaffold. The second component is nano-bovine serum albumin, or NBSA. Bovine serum albumin is a common, well-understood protein. The researchers transformed it into nanoparticles, vastly increasing its surface area and creating numerous active sites. These protein nanoparticles are the secret weapon. They are woven into the alginate scaffold by the gamma rays. The result? A powerful adsorbent material, a highly specialized nanosponge. The alginate provides the structure and stability, while the NBSA nanoparticles act as specific sticky traps for antibiotic molecules. When this nanocomposite is introduced into contaminated water, The antibiotic molecules are drawn to the NBSA due to specific chemical attractions, and they become physically entrapped within the alginate matrix. It's a sophisticated system designed to seek out and capture these specific pollutants with high efficiency. The team's experimental process was meticulous. First, they synthesized the nanocomposite by dissolving sodium alginate and NBSA in water and exposing the solution to a precise dose of gamma radiation from a cobalt-60 source. They then rigorously characterized the new material. Using techniques like Fourier transform infrared spectroscopy, they confirmed that the components had successfully bonded. With powerful tools like scanning and transmission electron microscopy, they were able to visualize the material's porous, nanostructured surface, confirming they had built the molecular trap they designed. Then came the crucial test. They prepared water samples contaminated with a common and potent antibiotic, ciprofloxacin. Upon introducing their nanocomposite, the results were remarkable. The material demonstrated an extremely high adsorption capacity, removing over 90% of the antibiotic from the solution in a matter of hours. The team also investigated optimal conditions, finding the material worked best at a neutral pH. Perhaps most importantly for real-world application, they studied its reusability. They found that the trapped antibiotics could be washed off the nanocomposite, which could then be reused multiple times with only a slight decrease in efficiency, making it a potentially cost-effective and sustainable solution. So, why is this so critically important? Residual antibiotics in wastewater are a growing concern worldwide. When we take antibiotics, our bodies don't fully metabolize them. The excess, along with improperly disposed of medications, enters our sewage systems. Conventional wastewater treatment plants are not designed to remove these complex molecules. Consequently, low concentrations of antibiotics are continuously released into our rivers, lakes, and oceans. This creates the perfect breeding ground for antibiotic-resistant bacteria, or superbugs. The constant exposure kills off susceptible bacteria, leaving the resistant strains to thrive and share their resistance genes. This process undermines the effectiveness of the very medicines we rely on, posing a grave threat to global public health. Furthermore, these antibiotics can disrupt fragile aquatic ecosystems, harming beneficial microorganisms that form the base of the food web and impacting the health of fish and other wildlife. For a country like Egypt, whose lifeblood is the Nile River, the implications are even more acute. With a growing population and increasing water stress, protecting the quality of every drop of water is a national priority. This research, developed by Egyptian scientists at a national institution, offers a potent, homegrown solution to a global problem with local urgency. It provides a potential pathway for upgrading wastewater treatment facilities, particularly those downstream from hospitals and pharmaceutical plants, to intercept these pollutants at the source. By developing these eco-friendly, highly efficient nanocomposites, this research opens the door to safer, cleaner water solutions. It's a step towards a future where we can enjoy the benefits of modern medicine without compromising the health of our planet. The future for this technology is bright. The research team notes that the next logical steps involve scaling up the production of the nanocomposite and conducting pilot-scale studies using real wastewater effluent, not just lab-prepared samples. They also aim to test its effectiveness against a broader spectrum of pharmaceutical pollutants. This study beautifully reflects the crucial role of interdisciplinary science, blending radiation technology, polymer chemistry, and environmental science, in solving our most complex challenges. But the responsibility doesn't end in the lab. It extends to all of us. We can contribute by using antibiotics only as prescribed, never flushing unused medication, and advocating for policies that invest in advanced water treatment infrastructure. Supporting and celebrating science that protects our shared resources is one of the most important things we can do for future generations. Stay with us on For Science Salah Lotfy channel as we continue to explore the groundbreaking scientific research that is shaping and saving our world.
Welcome to For Science with Salah Lotfy, where we explore the scientific breakthroughs shaping our world. Today, we're tackling a critical yet often invisible environmental crisis. It's a threat flowing silently through our rivers and streams, originating from a source meant to heal us. We'll be introducing groundbreaking research that addresses one of the most pressing global challenges, the removal of antibiotic pollutants from our vital water systems. So, how does this contamination begin? Antibiotics are essential in medicine and agriculture. However, a significant portion of these drugs isn't fully metabolized and is excreted into our wastewater. From there, they journey into rivers, lakes, and groundwater. These chemical compounds are designed to be stable, so they don't easily break down. They persist in the environment, creating a continuous, low-dose exposure with profound consequences. This persistence poses a serious environmental and health risk, fueling the rise of antimicrobial resistance, or AMR. When bacteria in the water are constantly exposed to antibiotics, the susceptible die, but the resistant ones survive and multiply. This process effectively creates superbugs immune to our life-saving drugs. Antibiotic residues in wastewater contribute directly to this silent pandemic, contaminating ecosystems and threatening public health on a global scale. The challenge is that our conventional wastewater treatment facilities are not equipped to filter out these resilient molecules. Therefore, developing efficient, affordable, and eco-friendly solutions to remove these pollutants has become a top priority for scientists worldwide. The search is on for a technology that can protect our water without causing further environmental harm, ensuring the safety of our ecosystems and our future. The silent creep of antibiotic pollution into our global water systems represents a pressing environmental and public health crisis. Released from pharmaceutical manufacturing, agriculture, and households, these persistent compounds often bypass conventional wastewater treatment. Once in our rivers and lakes, they foster the development of antibiotic-resistant superbugs, undermining modern medicine and posing a direct threat to ecosystems and human health. In the face of this challenge, a groundbreaking study offers a beacon of hope. Published in ChemistrySelect in 2025, the research is titled, Adsorptive Removal of Residual Antibiotics Pollutants from Waste Solutions Using Gamma-Induced Preparation of Nanocomposites Based on Sodium Alginate and Nano-bovine Serum Albumin. This work presents a tangible and highly effective solution. The core of this innovation lies in its unique synthesis method, gamma irradiation. This advanced technique uses high-energy rays to build the nanocomposite structure. Unlike traditional chemical methods, gamma irradiation is a clean process. It requires no harsh catalysts that could become secondary pollutants, making the approach inherently greener. It forges incredibly strong and stable bonds, creating a robust material designed for a difficult job. The researchers ingeniously combined two key biocompatible materials. The first is sodium alginate, a natural polymer from brown seaweed. It's abundant, biodegradable, and provides the perfect structural scaffold. The second is nano-bovine serum albumin, or NBSA. This protein is pivotal. Its complex structure offers a multitude of active sites that act like molecular magnets, attracting and binding to antibiotic molecules. Using it in nanoparticulate form exponentially increases the surface area available for this crucial adsorption process. When combined using gamma irradiation, these components form a highly porous, three-dimensional nanocomposite network. The resulting material is more than the sum of its parts. It demonstrates a remarkable synergy, leading to enhanced adsorption efficiency and a high capacity for capturing contaminants. This innovative approach, blending natural polymers with advanced technology, paves the way for a new generation of water purification systems to tackle the challenge of antibiotic pollution. At the forefront of scientific innovation in the region is the Egyptian Atomic Energy Authority. Within this institution, the National Center for Radiation Research and Technology is dedicated to harnessing atomic energy for peaceful purposes, including environmental protection. It is here, within the specialized Polymer Chemistry Department, that a critical breakthrough in water purification was born. This department focuses on pioneering the use of radiation to engineer advanced polymeric materials, creating novel solutions to some of the world's most pressing environmental challenges. The study was carried out by a distinguished team whose work exemplifies the center's mission. Their collaborative effort was spearheaded by four key researchers: Associate Professor Dr. Shaimaa Mounier Nasef, Dr. Mai Mostafa Mohamed, Professor Dr. Ghada Adel Mahmoud, and Professor Dr. Tarek Mansour Mohamed. This team represents a perfect synergy of experience and innovation. The senior professorship provides seasoned guidance and strategic oversight, while the dynamic, hands-on expertise of the other researchers brings theoretical concepts to life at the laboratory bench. Their collective expertise in polymer chemistry and radiation technology was the essential catalyst for this success. By combining their knowledge, they have forged a powerful link between fundamental science and tangible, practical applications. This research is more than an academic exercise. It is a direct response to the global crisis of water contamination. Their work provides a scalable, cost-effective model for removing hazardous antibiotic residues, safeguarding public health, and protecting fragile aquatic ecosystems. It stands as a testament to how focused, collaborative research can generate solutions with a truly global impact originating from the heart of Egypt. This study not only advances environmental cleanup methods but also showcases the potential of radiation-induced nanomaterials in solving real-world pollution challenges. It represents a step towards safer water resources and a healthier planet. Stay with us on For Science Salah Lotfy channel as we continue to explore research shaping the future of science and technology. Hello and welcome to the For Science Salah Lotfy channel. In today's episode, we explore an important research study that highlights the use of radiation chemistry in addressing one of the world's pressing environmental challenges: antibiotic pollution in wastewater. The research is titled Adsorptive Removal of Residual Antibiotics Pollutants from Waste Solutions Using Gamma-Induced Preparation of Nanocomposites, based on sodium alginate and nano-bovine serum albumin. This work has been published in ChemistrySelect under Chemistry Europe, with the reference ChemistrySelect 2025-10 E202406060 and copyright 2025 Wiley VCH GmbH. The study introduces an innovative nanocomposite material prepared by gamma irradiation, combining sodium alginate and nano-bovine serum albumin. These materials demonstrate a remarkable efficiency in adsorbing and removing residual antibiotics from wastewater solutions. The importance of this lies in combating water contamination, preserving ecosystems, and safeguarding human health from the long-term risks of antibiotic resistance. At the end of this episode, it is my pleasure. I am Professor Dr. Salah Lotfy, professor at the Egyptian Atomic Energy Authority, to extend my deepest gratitude and appreciation to the distinguished researchers for their outstanding efforts in producing this valuable work. Associate Professor Dr. Shaimaa Mounier Nasef, Polymer Chemistry Department, National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority, Cairo, Egypt. Dr. Mai Mostafa Mohamed, Polymer Chemistry Department, National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority, Cairo, Egypt. Professor Dr. Ghada Adel Mahmoud, Polymer Chemistry Department, National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority, Cairo, Egypt. Professor Dr. Tarek Mansour Mohamed, Polymer Chemistry Department, National Center for Radiation Research and Technology, Egyptian Atomic Energy Authority, Cairo, Egypt. Finally, I also want to thank all of you, our valued viewers and followers of the For Science Salah Lotfy channel. Your engagement means a lot, and I warmly invite you to share your thoughts and comments about this research in the section below this video.